When I joined CU Boulder in the Department of Biochemistry in the summer of 2015 as an assistant professor, I was lucky to accept two Ph.D. students into my lab, Nick Hill and Patrick Thomas. They will debate as to who is the first student, but they probably started about 8 minutes apart, just like the identical twins that I had shortly after starting my lab, disrupting my life quite a bit. Nick and Patrick were instrumental in helping me build my lab group and culture, and I'm greatly appreciative. Here I'll tell you about Nick. He came to my lab eager to do synthetic biology and was always interested in an industrial path after grad school. I taught him in core and knew he was super smart, so wanted to challenge him. I was told at a talk that a grad student could not complete the ambitious project that I had proposed. This cost me my backup job. I knew this was the project for Nick. Here is the premise of his thesis. Populations of cells are heterogeneous, so one must consider the single cell. However, the carbon-fixing organelles within a single cyanobacterial cell could also exhibit heterogeneity. Thus, it would be advantageous to study a single organelle in a single cell. This required Nick to develop a synthetic biological system to control the assembly and dilution of carboxysomes in the population, film them using time-lapse microscopy, and learn how to code to analyze the data. This included the development of a novel algorithm to measure the activity of a single carboxysome for its entire life. The implications of this, the first method to measure the activity of a single carboxysome in a living cell, are enormous. These structures are responsible for fixing 30% of global carbon dioxide. So understanding the structure and function is crucial to enhancing biological carbon sequestration into food, fuels, chemicals, and even biological building materials. Nick's research has been published in top journals, and a review from one of his manuscripts entitled "The Life Cycle of a Carboxysome" stated, "The writing is essentially without flaw and should be published as is." And that was a great honor to receive a review from that on a work that was largely written by Nick. Nick has been a great mentor in the lab to both other grad students, high schoolers, and undergraduates. And through his willingness to collaborate and his ability to do excellent, high quality, rigorous work, he's contributed to many other projects in the lab. Nick is now heading off to industry to follow his plan. He will be missed, but his contributions will be long lasting in the lab and the field. Thanks, Nick, and congrats.